Hi, my name is Ella Williams, and today I'm going to be talking about Duchenne muscular dystrophy, also known as DMD. More specifically, I'm going to be talking about how the immune system reacts to the gene therapy that has been developed for this, um, and how the T cells can actually fight this protein. So, what is DMD? According to the Muscular Dystrophy Association, DMD is a genetic disorder characterized by progressive muscle degeneration and weakness due to alterations of a protein called dystrophin. This is what helps keep your muscles intact. So over time, we see a loss um, in muscle abilities. We lose the ability to walk, and just overall function deteriorates. So some basic concepts. How this works. Duchenne muscular dystrophy is a severe X-linked disorder, so this means we see it predominantly in males, caused by a mutation in the DMD gene. This can lead to the absence or just alteration of dystrophin, which is crucial for muscle stabilization. There are gene therapies on the market that use adeno-associated viruses, or also known as AAV vectors. It delivers a shortened version of dystrophin called microdystrophin, which you'll hear me talk more about later. Um, However, there's a significant barrier to this as the body can attack these microdystrophin proteins. For someone whose body has never had dystrophin or has a different form of dystrophin in their body, it can be targeted as foreign, and so some of these therapies haven't worked because of this. So here's a quick drawing of how this can work. So first we see the AAV vector in the top left corner there, and it holds the DNA for the microdystrophin, which is then inserted in the nucleus where DNA becomes available for transcription. And then as we see, it goes on to be translated and eventually turn into a protein. These peptides are secreted and picked up by an APC cell, or sorry, an antigen presenting cell, also known as APC, and presented to T cells, which can then target the cell and cause cell death. So, a quick poll everywhere question. Why do you think gene therapy for DMD focuses on microdystrophin versus the whole dystrophin protein? Well, to answer that question, first of all, what the heck is an AAV? An AAV, or adeno-associated virus, is a commonly used viral vector that is used to deliver gene product for gene therapy. Um, However, for an AAV, the viral load is very small. So the dystrophin protein is a fairly large protein, um, which the full DNA content wouldn't fit into these viruses. So, in order to make this work, they have to use microdystrophin. So they targeted parts of the gene that are more important for function and are able to replace the effects of the full dystrophin protein most effectively, um, just so that they can fit it into this small capsule. So next I'm going to talk about some figures from the papers that I looked at. Firstly is from this Mendel paper. This is figure 1 talking about CD8 and CD4 T cell responses to the microdystrophin peptides. I want to focus mainly on panel A and the flow cytometry chart in panel C. Um, so firstly, the y-axis is going to be measuring T cell activity, and we're looking specifically at peptide 19. The x-axis is different APCs for the peptide. So it's essentially what is presenting these antigens to the T cells. How it's presented is what shapes the response from the T cell. So we see a reaction in C and T. So it shows how strongly T cells respond to peptide 19 when presented by different antigen-presenting cells. And then MDP1 was a positive control that's essentially just a solution. I have a help slide. It's a synthetic peptides. It's kind of the solution that it was sitting in. And then this will come up later, but HLA is the human leukocyte antigen system, and it's a complex of genes on chromosome 6 in humans that encodes cell surface proteins responsible for regulation of the immune system. This may sound familiar because it's essentially the human version of MHC. The main difference between HLA and MHC is that HLA, the human leukocyte antigen, is a specific type of MHC found only in humans, where MHC is more general. But to go back, the T cells only responded when APC C and K are used. This must mean that C and K must share an HLA class 1 molecule capable of presenting peptide 19 in the way that T cells, specifically CD8, can recognize. This means that there is some form of HLA restriction going on. The T cells are restricted to recognizing peptide 19 only when presented by that shared HLA type. They get a little bit more specific in a different panel, but essentially, HLA class 1 cells activate a specific T cell. And then a poll everywhere question. I kind of already gave this away a little bit, but what type of cells do you think will be activated by peptide 19? And just a little hint, HLA B cells, which was specified in panel A, is an MHC, that should say MHC, class 1 molecule, class 1. And the correct answer is CD8 because we know that CD8 and MHC1 go together. And HLA is B is classified as a class 1 MHC molecule, which only presents antigens to CD8 T cells. These are the antigen presenting cells, remember C and K, that express this specific HLA could stimulate T cell, and IFNY production is just a marker for activation. And then panel C, this is a similar thing, but for a different peptide within the microdystrophin or within the dystrophin gene, or protein, sorry. And it's measuring what kind of T cell, CD4 or CD8, is responsible for the immune response to peptide 23. So we can see on the flow cytometry chart, we see it could either be both CD8 and CD4, just CD4, just CD8, or neither. And there's a clear winner. It is only CD4. And then total was the positive control. So we see that peptide 23 is activating a different form of T cell than peptide 19 was, which we saw in panel A. We see that there. And another poll everywhere question, and again, my link is at the bottom, and the question should be open. But what conclusions can be made from this data? You can pause. But it was a select all, so answer number 1, since CD4 T cells respond and CD8 do not, peptide 23 must be presented by MHC class 2 and not class 1. Because again, we know that class 2 goes with CD8, class 2 goes with CD4, and class 1 goes with CD8. So responses are HLA restricted, meaning that effective activation depends on the compatibility between T cells and the presenting MHC molecules. And different peptide fragments from the same protein can activate distinct T cell subsets depending on how they are processed and presented. And then some implications. AAV-mediated gene therapy, which is what this is, can activate the immune system with antigen-presenting cells displaying microdystrophin peptides on MHC1 and 2 and can trigger CD8 and CD4 T cell responses, immune responses against microdystrophin, or the AAV capsid itself can lead to destruction of these muscle cells, which basically eliminates the effectiveness of these therapies, and can raise a lot of concerns for patients. And then just to wrap everything up and kind of summarize what we've talked about, Duchenne muscular dystrophy is a mutation that causes a lack or a change in function of dystrophin, which is integral for muscular integrity. The gene therapy that is currently available is a microdystrophin, so just part of the protein based on studies that show which parts are effective, which ones are activating T cells, which ones are not. And it's delivered with an AAV vector. Unfortunately, now they are activating the immune system through, as we talked about, and make the certain presenting molecules. There is hope for the future. Future therapies have to account for these immune risks by screening for T cell reactivity or even using immunosuppressants. This can be a little bit risky, but they're hoping for more developments on this in the future. Thank you. These are some sources.